Hi everyone. In this video, we'll look at the replication of the lagging strand, and we'll take a quick look at the final steps to complete the newly replicated DNA. Remember from the previous videos that the lagging strand is the side of the template molecule that has the replication fork moving towards its 3' end, so that the 5' end of the new daughter strand is towards the fork. This makes replication more complicated since DNA polymerase can only build 5' to 3'. So we're going to have to effectively build the lagging strand backwards in the opposite direction of the movement of the replication fork. To do this, the lagging strand will be synthesized in short sections called Okazaki fragments. Each one of these fragments will be synthesized 5' to 3' from its own primer. We'll start here with primer 1, which DNA polymerase 3 can use to start a new DNA strand, bringing in one nucleotide at a time, 5' to 3', just as we saw with the leading strand, until the first Okazaki fragment is complete. After this, the second primer can be brought in, and DNA polymerase can move closer to the replication fork to start building the next fragment. The process will continue in this way, with a new primer for each fragment, until the end of the template is reached. Now we have a new double-stranded DNA molecule, but it's full of short pieces of RNA which need to be removed and replaced with DNA. This is carried out by another enzyme, DNA polymerase 1. Now, DNA polymerase 1 has 5' to 3' exonuclease activity, which means that it can cut out nucleotides and replace them in a 5' to 3' direction. DNA polymerase 1 is going to move down the DNA strand, cutting out the primers, shown here in red boxes, and fill in the gaps with DNA nucleotides. You'll see that once this is completed, we have another problem. When nucleotides have been replaced, we have gaps in the sugar phosphate backbone. This is where DNA ligase comes in. It will act like molecular glue, forming phosphodiester bonds between the nucleotides, sealing up the backbone. Now, while we're only looking at the lagging strand here, remember that this also occurs in leading strands, just to a lesser extent, since the leading strand only has one primer to replace. Thanks for watching, and if you haven't had a chance yet, please go check out the other videos on DNA replication.